All right, welcome back, everyone. We are now ready to move into session 2 of our WeedPedia training with Koen Nijbroek from KeyGene. So again, please, if you're online, you can type your questions into the Q&A box. And we will be, in this section, taking a look at some more intermediate functionalities within Pedia. So Koen, I'll turn it over to you. Thanks.
Thank you. So yes, we are now going to use Pedia with a very specific scientific research topic in mind that will hopefully give you an illustration of how you should use Pedia and how to narrow down on all the information that you may have. So in this case, we'll be looking at rice, and a very important phenotype of rice is, of course, yield. Especially the number of panicles and the number of grains per panicle that a certain plant can have slash carry, of course, is heavily involving the yields that you will get out of this specific rice plant. Therefore, rice plants with larger branches, more branches, or more number of spikelets are very beneficial for the yield increase. So we'll be looking at the component yields and see how we can use Pedia to find the causal piece of information that generates this preferred phenotype. So a major step we won't do in this demonstration. So usually when you have a phenotype and you want to find the causal gene or function of that, you need to find genetic markers that are close to this region of interest. In this case, the markers were known based on literature. But of course, when markers are not known, then you need to involve a major step to get closer to your potential QTL for the trait that you're looking at. So in this case, based on literature, Ashikara et al., we knew that a particular QTL named GN1A, which stands for grain number 1A, harbors a very positive phenotype regarding an increased yields. And two flanking markers were known, R3192 and C12072S, which for the rest of the talk I will just refer to as the left marker and the right marker. And in total, the distance between these two markers is approximately half an MB or 404 KB. So there is a tremendous amount of genetic and genomic information within this region. And we will use Pedia to display the information on this QTL. And the strategy for doing that is by first finding the markers based on a simple keyword search to see if these markers are already present in the Pedia database. And if they are present, then they will prompt you with a genomic locus. And if they are not present, so for example, the data of that particular paper has not been incorporated into Pedia, then you can use a BLAST to align the sequence of these markers and then find your genomic coordinates. We will then zoom into this region in the genome browser and basically activate all the data sets that we deem interesting in order to continue with the research. Obviously, that's the gene passports. That's the most essential piece of information if it's available. And for rice, there's a lot of public information known from Ensemble. So there's a lot of data tracks in the RicePedia from public data. And then we will browse through the gene models and basically decide on all the information that we see, whether these genes are potentially interesting for the phenotype that we want. And then we'll make a top list or top potential candidate genes that cause this desirable phenotype. And after this, there's obviously a lot of testing involved to see whether these genes that you find in silico are actually indeed causing the phenotype. So we'll do that by going to portal.koepedia.com. So that's not your WeedPedia. This is the cloud-based Pedia for our shareholders and third-party customers. And basically by using a tenant ID, you will only see the data that's relevant for you. So in this case, I will use the demo tenant, and the demo tenant only has access to rice. So to start from scratch, when we have this big research topic, best practices learned that you might want to make a workspace in order to save all the information that you find interesting. So I will make a new workspace for this example, call it Rice 2. We go to Rice 2, and I will add some text to this. And this serves as the starting point for our expedition. So the first step of the strategy was to enter the markers in Pedia and see if your data is already present in the database. So one of the flanking names was called R3192. And if we do a search, then you will see that there is data in the database that contains this specific keywords. And we have no gene passports available for this keyword. That's logical because the keyword that we entered was a marker and not a gene. However, we have genomic features available, and you can already see some of the description there. So it's related to a QTL, and it was obtained from the Grameen database. And we also have a restriction site hit available. And you see that these are hits for 3 different genomes. We have 3 genomes of rice available in this Pedia. However, we will only focus on the Japonica in this example. We also have genetic maps for rice. So markers, this marker is available in certain genetic maps. And you can see here that all the search terms that we, or all the search results that we obtained are all located on chromosome 1 for all 3 genomes. There's no pathway information available, and there's also no PubMed information available because the outside link for this demonstration Pedia has been disabled. So we can look at one of the results that we found. And then it will bring us to the feature information. And if I click here, then it will also bring us to the genome browser. And we have not enabled any of the tracks so far. So what I will simply do is enable the left flank. So this is the other way of doing it. What we just did right now was searching on the keyword and we found the results. However, if that keyword could not be found, then it's also possible to do a blast, and this is the outcome of that blast result. So I can right-click on this feature and say add it to the workspace. And then it will prompt me, which workspace do you want to use? Well, I have 3 workspaces, but in this case, we will use RISE2. That's the one I just made. Feature save to workspace. You can now close this window. So now we have the left marker stored to our workspace. We can also do that for the right marker, of course. And then we have to type in the other one, C12072S. See if we find any results there. And here we only find 2 of the genomes. So if you would have been working on that other genome, you would only observe one of the markers and not the other one. It doesn't mean that it's not available. It simply means that the data underneath this reference genome does not contain this search term. So if we would have continued with the other genome, we would have to blast the right marker in order to find it. But for Japonica, which is the genome that we will be focusing on, we did find the results. So again, I will navigate to this region. Then we end up in the genome browser. And here is the blast track that I also made. And this is something that we can save to the workspace. The highlight is not a data, it's just a feature. But if you would enable all the tracks, then you would find the result of the search as well. But in this case, saving the blast result is a bit quicker. Again, add this to the workspace, RISE2, close the window. And now we go to the workspace. And now you see that your starting point of the research topic now contains 2 data properties from Pedia, the left marker and the right marker. So that means that from now on, we can visualize everything that's between these 2 markers. So we can browse to this location. This is the left marker, and this is the right marker. And here we had the left marker again. So this was the left marker, this was the right marker, but now we want to see everything that's in between these 2. So I will just copy the leftmost coordinates and replace it with this one, and to go. And now we should have the whole QTL in once. So you can see it here, the left marker is located here, the right marker is located here. So in between these 2 markers is the potential gene of interest within this QTL. And by enabling the gene passports, which were also present for the 2 genomes in the WeedPedia, you already have a bunch of information available. And if I would highlight this region, there you can see that we have around 50 genes in this QTL, which may cause this particular trait of interest. Besides the gene passport, you may also want to have additional information, such as, in this case, the genes from ensemble. And then all of a sudden you see that your window that you are looking at is getting bigger. And the more data you add to the view, the less or the more difficult it becomes to interpret all the results. And in order to narrow down on this search, there is no other way than going to each and every single data point that you have within this highlights, because in theory, every single data point that you see in this yellow box may contain something relevant. And it doesn't mean that by enabling only a single track, you will find the piece of information that you are looking for. It may very well be that it's not present in the gene passports for whatever reason, that the extensive genome annotation was not able to pick up this gene. And it might be that public information does contain the information that you were looking for. So if we narrow down the window a little bit by simply removing some of the metadata, you can already see that there is a tremendous amount of differences between these two data sets. Both contain genes. However, the ensemble gene database contains a lot more information. So if you would only look at the gene passports, you miss half of the information, at least, that is present in the ensemble information. And then for rice, there's a lot of other relevant tracks as well that you may want to involve. For example, gramine, something that I also mentioned earlier, a very extensive database, very well suited for rice. But again, if we would change the view of this one, you will see that this one is even more limited. So therefore, the more data you have in your system, or basically the more relevant data you have in your system, the more it will, in the end, help find out the question that you have. And of course, it may also very well be that even within this view, with the amount of data that we have here, you won't be able to find anything interesting. And then, of course, you need to do more experiments, check the information that you're working with, and then try again. So the more data you add to the system, the better you're able to do your research, but also the more complicated it gets. So yeah, you have to be careful when browsing through Pedia and what types of information you want to display. So if I would start with the gene passports and go to the normal view again, I would just have to go to each and every one, click on the gene passports, and then based on the information that I see here, decide whether this particular data point is of interest to me, yes or no. And if you have some more background information on the research topic that you are working on, then the predicted localization or the Interpro results or the Go accessions or the clustering or the BLAST or the RNA-seq, whatever can help you in deciding whether this is interesting, yes or no. So this particular gene seems to be working with replications of cells. Well, that doesn't sound directly to me related to increase of yield. So in this case, you would simply close this data point and go to the next one. And then again, look at the gene passport, look at all the information that you have, and decide whether this is of interest, yes or no. And this is a tedious process because there's a lot of data in there, and Pedia will not directly give you the answer. It's basically you who need to find that answer by the means of Pedia. And one of the things that may help is variation data. This is something that was not available for the WeedPedia yet. So imagine that you have a set of rice samples that show this particular phenotype versus a set of rice samples that do not show this particular phenotype. And for good reasons, you believe that this phenotype only relates to a single gene and is likely a loss of function of this gene. Then you can simply have a look at the variation data for each and every single gene and see if you find a potential variance that correlates well with the sample groups that you defined. And if you see such a variant, you might think, well, this is something that may cause this particular phenotype. This is something that I want to store. So I will add this particular gene to my workspace, close it again, and go to the next one. And so you go through each and every single data point and basically decide whether this is interesting, yes or no. And you simply do that by looking at all the metadata that is available, gene passports. If gene passports are not available, then you have to rely on other types of public data. You might even want to do some blasting to further narrow down this region if you have other information. But in this case, in this demonstration, it doesn't make sense to go through each and every single gene because that takes way too long. But we found a very interesting gene in the Gramine dataset. And I will highlight this particular gene only. So that's CKX2. And you see already, well, the view is a mess. So if I want to highlight this, then I need to collapse this a little bit. And basically what I want to do is only highlight this area. Highlight. We will enable all the normal views again. And again, it looks like a mess, but as soon as you zoom in, as soon as you lose the remainder of the context that you do not want to look at, all of a sudden the view gets clear. And that's the way you want to work in a genome browser. You start from a very large area and you narrow down to a smaller window every single time until the data becomes understandable. So now we have, again, these three tracks enabled. We know from our extensive search that this is a very interesting piece of information. And you see already that it overlaps pretty well with the other two data sets that we have. So we have multiple sources of information that we can have a look at. Well, the Gramine information is public data, which is often very limited. So if we have a look at what this feature tells us, it only tells us that it is a gene. It only tells us where it is, how long it is, and what the sequence is. There's no description or annotation present whatsoever. But if we look at the other track from Ensemble, we can already see that there is some more information available. Apparently, the description that Ensemble gave to this gene is similar to cytokinin dehydrogenase 2. Also doesn't say us that much, but there are switch spots accessions mentioned. So you might want to search up this specific identifier on the UniProt switch spots and find some more metadata. And then we come to the most valuable source of data in Pedia, if you have it, and that's the gene passport. So that's everything that public data cannot offer. You really need to do extensive genomic annotation for this. So if we have a look at this particular gene, we see that the predicted localization is all over the place. So this doesn't help us very much in what it is. If we go to Interpro results, we see that based from other public data, we get some keywords that tell us a little bit of what the function may be. And one of the things that's raised our suspicion is cytokinin, because cytokinin is a growth hormone. So that makes sense when you're looking at the trait yields. And if we go to the BLAST results, we also find some information from BLAST, and again, cytokinin. And it even mentions the QTL that we were looking at. So this is a very promising candidate that may yield this particular phenotype. Also in the Go accessions, again, cytokinin is mentioned. So multiple sources of information tells us what the likely function of this particular gene/protein is. And therefore, we deem that this is very interesting. There's even a phylogenetic tree, which is really extensive, that can also help in judging information. And again, variation information. So again, if there's a particular variant in this gene, which based on function is a promising candidate, then all of a sudden it becomes very interesting. A new type of data pops in, which was also not available for the previous gene, is RNA-seq. So if you have RNA-seq experiment data added into Pedia, and this gene is mentioned in there, and there is expression data available, it will be automatically shown in the gene passports as well. So imagine that there are lines available where this gene was completely knocked out, then you would not see any expression at all, etc. So again, we will save this to the workspace. Rise to success. So imagine that we went through these 150 data points that we saw within our QTL. Imagine that you saved 10 potential candidate genes to your workspace. You will find out that if you, for whatever reason, for example, close Pedia or click away a window that you did not want to click away, that you can immediately go back to the genes that you saved as interesting. So this was the most promising candidate gene for us. And we immediately come back to the window that we also saw when going through the genome browser for the genes one by one. So we went from literature to 2 candidate regions or 2 flanking markers to a QTL to a list of genes. So you see, we start from a very big window and then we slowly move to smaller and smaller areas. And now, ideally, we also want to find variation in this gene that may explain this phenotype. So if we would go to the and now my screen is frozen. There we go. And we go to Japonica. Then we have a whole number of accessions resequenced. So there is a phenomenal public available data set that contains 3,000 individual rise lines that have been resequenced in a very high depth. And these rise lines are pretty much obtained from all over the world. And you know from research which lines contain, for example, this very desired phenotype and which don't. So then you can say, well, I want to enable this one, I want to enable this one and this one. So I will select 4 lines, apply the selection, and I will look at the variant data. And then you will see that apparently for the first line that we have a lot of variation present across all the chromosomes. But for the other 3 lines, we only have information available for chromosome 1. Well, luckily, chromosome 1 was also the chromosome where this gene of interest was located on. So you could simply go to chromosome 1 and then look for patterns in the particular gene that you are interested in. So if we would get the coordinates of this gene again, and then paste it right here, then you see that we find 2 SNPs. That's all, or 2 blocks which contain 3 SNPs in total. There are 2 lines that do not have any SNP at all, and there are 2 lines that do contain information. Well, you can export this to a VCF file, except not in this demonstration because that has been enabled. And in the VCF file, you can then look in detail what alleles were present, what functional annotation was present, etc. So then all of a sudden we go from 2 flanking markers in the QTL to a gene to potential variation of interest. And then of course comes the validation afterwards that needs to be done outside of Pedia, so wet lab experiments to really verify whether your potential candidate list contains the gene that is causing this phenotype. So switching back to the presentation, we found one of the genes that was coding for cytokinin. It's a growth hormone that promotes cell division, and it is now known to influence various aspects of plant growth, such as seed germination, leaf expansion, and reproductive development. And it also controls, as mentioned, cell division and lateral meristem activity. And the theory is that a accumulation of cytokinin in the meristem could explain the significantly increase in grain numbers that were obtained from these preferred plants. So it turns out that after doing some extensive research on these genes and lines of interest, that a 11 base pair deletion led to a premature stop codon, and therefore this gene contained a loss of function. And this loss of function was later, by validation, indeed determined to be the causal variant for the observed phenotype. So then you might wonder, now that I know this, now that I went all the way from this massive region with this massive amount of data to a causal variant, which other lines or accessions or crops or different genomes also contain this apparently preferred phenotype? And then again, you go back to Pedia to have a look. So you see that it's a back and forth working between data generation, Pedia, validation, more data generation, Pedia, etc., etc. So in this whole story, Pedia is every time used as visualization and interrogation to basically make sense of all the data that you produce. And we showed this already. So this is an example of how to operate Pedia when you have a very specific research topic. And I mean, now I show this in a half an hour approach, but this took researchers more than a couple of years. So it's a slow and tedious process to gather information, to interpret results, and to finally find that piece of information that is relevant. So hopefully this gives you a bit of insight on how to use Pedia and how all the different aspects come together. And with that, I'm happy to answer any more questions if there are any.
All right, we've got the opportunity now for questions, and thanks, Carlos, to come around again with the microphone. But thanks, Koen. I mean, such a great example, and you can just imagine, you know, the various things that people might be looking for in weeds. Now to be able to go through that same process, conduct a QTL, find markers, zoom into that region, look at the genes that might be involved, their annotations, and think about your hypothesis for which genes might actually be controlling it. It's all possible now, so it's just tremendously exciting.
Yeah, I mean, it's the way that one would normally work as well. Yeah, but now in just one system. That's the whole idea behind this.
Yeah, it really opens up the non-model plants, right, to this kind of inquiry. So again, if you're online, virtual, you can use the Q&A box to type in a question, or any questions here in the room. Okay, here we go. Yeah. Just getting the microphone to the question asker here, Koen.
Hi, good morning. I just wondered, do you have anything similar in any polyploid species? I know you showed for rice, just wondered.
Yeah, so rice is one of the most easy crops that you can probably do research on. It's diploid and often highly homozygous. So depending on the nature of the crop that you're looking at, things can get very difficult, and for many weeds that's the case. We do within KeyGene a lot of polyploid research as well, and we also use Pedia for that. But in the case of polyploids, you basically need to decide whether you want to work with a haploid representation of your genome and then present all the data of multiple alleles to that particular haploid representation, or whether you are able to individually obtain the chromosomes of each allele and then do your research on that level. So it depends on how much knowledge or how much data you were, or how much data you have obtained successfully in order to really do research on polyploids. And yeah, it's complicating, especially considering the different levels of autoploidy or alloploidy. But still, the same core functionalities of Pedia apply to those cases as well. And what's nice to mention is that we are also working on pangenomic visualization. So that goes from a single representation of a genome to a graph representation of multiple genomes, maybe in the future even all the wheat genomes that you may get your hands on. And that's also applicable for alleles. So then you can see the different regions that are unique within alleles, or the regions that are similar. You can see structural variation, etc. So polyploid research is complicated, also in Pedia, but there are certainly possibilities to do that.
Thank you.
Hello. So I have a question. So I use phytosome. It is similar to Pedia. So I want to know, are there any key differences that I can specifically use the Pedia rather than the phytosome?
Yeah. So the key differences are that phytosome is, of course, a single database maintained by a single group, and you cannot put your you can't put your proprietary information in there, but then it's visible for the whole world. And within Pedia, your proprietary data is protected, so it will not go online, and it will remain on premises or in the cloud, but accessible only for you. So that's one of the major differences versus a public visualization tool. And of course, all the data that is in phytosome, you can also, in theory, put into Pedia. And of course, we have functionalities available that phytosome doesn't have. We have an extensive variomics viewer, for example, that's quite unique compared to everything that's out there. So yeah, phytosome, I mean, is a amazing piece of resource. But if you want to add other resources in there, then things get complicated, and therefore Pedia comes into the picture.
Thank you. And of course, we don't have wheat genomes in the phytosome.
Yeah.
Thank you.
Sure.
So that's a good point to add, Koen. I think you're covering this in the third section. But for example, anyone here, once they have their WeedPedia account, say they have their own RNA-seq data set, and they'd like to put it in there to visualize it, but maybe it's not published yet or it's something proprietary. So when they upload it, they can keep it so it's only visible to them. Or if once they publish it or they do want to share it, they can then choose to make it available.
Yeah, that's true.
Right. We have a question from online from Tan at BSF. So the question is, with extensive Indica and Japonica genome information in RicePedia, is it still necessary to have red rice genome in WeedPedia? In other words, can the current rice genome information in RicePedia be substituted for the red rice genome of WeedPedia?
I'm not familiar with the red rice genome. So, I mean, these are two completely different Pedias. So the rice Pedia is really tailored to rice only, whereas the WeedPedia obviously is also tailored to wheat only. But that doesn't mean that you can make connections between them. So if you want to place information from one database to another, you need to do that by bioinformatic ways. But then you can upload again that information into KeyGene and in such a way make use of resources that are only available in other Pedias. Not sure if that really answers your question.
I wonder, Koen, it might be a nice example for the once the comparative genome viewer tool is in there, because I'd imagine in red rice, weedy rice, there's going to be structural variation. There will be lots of differences in regulation of genes. So maybe that's where having that genome comparison could be really interesting.
Yeah, I mean, yes, it works. If genomes resemble a form of similarity to each other, you need to make connections. And indeed, with the pan-genomic viewer, you can easily view connections between various genomes. It's also possible in the regular version of KeyGene that I already showed, but your ruler, we call it a ruler, the genome that you display your information to is in the current situation always a single genome. And in the future, you can look at multiple genomes at once, and you are not limited anymore to only look at one genome at once.
Yeah, that's great. That's an exciting thing. I think I'm curious what everyone will think about that, and at our discussion workshop on Friday, what ideas you might have as we move through having one reference genome for a given species to thinking about pan-genome genetic variation that's out there across the genome. Koen, we have another question from Ana Caicedo asking about the cost of KeyGene. Is it affordable for labs, or does one need institutional buy-in? And I guess I could take that from the perspective of as the International Weed Genomics Consortium. So we have it's a public-private partnership, the consortium. So there is funding from the founding member sponsors. So the companies you'll see on the slides going through Corteva, BASF, Bayer, and Syngenta, as well as Global HRAC with sponsorship, and then the Foundation for Food and Agriculture Research provided matching funding. So with that, one of our visions for the IWGC was to have and make available, user-friendly online genome analysis tools to help make it an easier entry to weed genomics for people to work with. So that's how we identified and worked with KeyGene to, so through the consortium, we're basically, is how we're covering the cost of the access to Pedia, and then this database development that we're going through right now. So I would say if it's something that a single lab wanted to look at, I don't know, maybe Koen, you can answer that if there are options. But for everyone who's working on weed genomics, you have access to Pedia. So we're almost there to where you'll have your login for this. We've been in a beta test phase to make sure these gene passports and everything is working in our first 2 genomes that are in there. But if you're at a founding member company, then you'll be able to get your login. And for everyone who's at academic partners who are members of the IWGC, then you can also have a login for WeedPedia and be able to access these tools for all the genomes. And then as I mentioned, the ones that we're doing, the 10 through the consortium, will come online after a year, and the ones that are already public then will be right in there. Is there anything you'd like to add to that, Koen? Is our feed frozen? Okay, you're back. Is there anything?
I'm back.
Yeah, sorry. Is there anything you'd like to add as far as the how people would access, or maybe Pedia otherwise if they're working on something not weeds?
Yeah, so it's in a sense fairly simple. The Pedia framework is the same for everyone regardless of the crop. So the only thing that an individual lab or institute or company has to do is to obtain a license from KeyGene, and there's a fee attached to that. For an individual, it's a lot. For a company, depending on the size, it's probably not that much. So obtaining a standalone version of KeyPedia is pretty cheap, depending on how much effort you want us to do to build your Pedia. So for example, if you want genomic annotation or genomes in there or publicly available data sets in there, then of course the price gets higher, but you can also do that yourselves. So it's a fairly easy thing to obtain if you want to.
Great. All right. Any other questions here in the room? All right. And I think we've gone through all the questions online. Well, thank you again very much, Koen. That was a great, I really like that session to kind of see, again, what's possible, and we can imagine how then our data sets and weeds will follow along perhaps similar stories. So yeah, I think that was a great demo. Just a reminder for everyone here in the room, again, if you're looking for charging or electricity access, we do have more chargers in these first couple rows. So feel free to move up in the next session if you would like. And then I think what we will do, we're pretty close to on time here. The break was scheduled for 11. So we're at 10:51, so we will come back at 11:15 local time. So we just have a little 10 minutes extra for the break. And at 11:15, then we'll conclude with the third session, which is kind of the most advanced use case scenario, right, Koen, where we'll be looking at supported data types and actually how you could upload your own data sets into the WeedPedia.
Yep. This will get into the details. Yeah.
Great. All right. Well, thank you so much. And we will be back here at 11:15 Central Time.